pathway, going to take a look in details on the C4 pathway. So one of the characteristics is they're going to be twice fixation of carbon dioxide. First fixation of carbon dioxide occurs at mesophyll cells, and the second carbon dioxide fixation occurs at bundle sheath cell. So first carbon dioxide fixation used PEP carboxylase as the enzyme. So the enzyme PEP carboxylase catalyzes the reaction between carbon dioxide and the carbon dioxide acceptor, which is known as PEP or phosphoenolpyruvate. Okay, phosphoenolpyruvate. Phosphoenolpyruvate contains 3 carbon, so carbon dioxide 1 carbon. So that's why the first compound or substance that is produced is oxaloacetate, which consists of 4 carbon. Okay, back to the PEP carboxylase. The PEP carboxylase has high affinity of carbon dioxide, even when the carbon dioxide level is very low during the hot and dry weather. This is one of the way of how the C4 pathway as well as CAM pathway minimize photorespiration, okay, because this enzyme can still fix the CO2. Unlike Rubisco, Rubisco cannot fix CO2 at low level, so that's why it only fixes the oxygen. And as a result, it causes the photorespiration instead of a Calvin cycle, and it cannot produce the sugar or glucose. It reduces the photosynthetic output for photorespiration. First cell, mesophyll cells, where the first carbon dioxide fixation occurs. So the first, we have the carbon dioxide acceptor, which is PEP or phosphoenolpyruvate. PEP consists of 3 carbon, and then adds with carbon dioxide, which consists of 1 carbon, and then it produces oxaloacetate with 4 carbon in the presence of PEP carboxylase enzyme. Okay, then the 4 carbon OAA or oxaloacetate will be converted to 4 carbon compound, which is a malate. And this process, this conversion of OAA into malate uses reducing agent, which is NADPH. Okay, so OAA to malate. It uses NADPH, which is gathered from the light reactions or light-dependent reactions. And then transported to bundle sheath cell. This malate is then transported via the plasmodesmata. So this is one cell, another cell. So there's a junction between the cells, which is known as plasmodesmata. So malate is transported to bundle sheath cell via the plasmodesmata. And there at the bundle sheath cells, malate is decarboxylated and forming pyruvate and release the carbon dioxide. Okay, so decarboxylation occurs. Carbon dioxide release, and then malate will be converted into pyruvate. Pyruvate, which consists of 3 carbon. Okay, you have carbon dioxide here, so this carbon dioxide will enter Calvin cycle and then produces sugar.